So today we're going to talk about macromolecules. So we talked about digestive systems. We have talked about, the hardware of the digestive system. So now we're going to talk about what's going on. What are we digesting? So we're digesting things of macromolecules. And we know everything that we kind of use is made out of atoms, whether that's food that we use, food that we eat, water that we drink, or tools that we use and the houses that we live in. They're all made out of atoms. And atoms made of molecules. However, molecules also make up macromolecules, which are bigger molecules essentially. So to first understand what's these molecules, we have to understand why they're joined together. So we talk about condensation and hydrolysis, which basically either combines two different stuff together by removing water, or you're separating two stuff then by adding water. So we have condensation up here. We have C. So C can be anything essentially, and then you have an OH group, and on the right you also have OH and C. So now you're removing water, so you're gonna get COC. So you have removed water out of this entire reaction, and by the end you're gonna have a brand new joined together molecule. And then below it we have hydrolysis. Hydrolysis is basically we're taking in water so we can break two things apart. So you can see that through the reaction. We have the COC and we add water in there. Now it's broken apart. So, however, one thing is that it must have at least one hydroxyl or carboxyl group. So let's look at macromolecules. So macromolecules are polymers. So polymers are large molecules that are made up of repeating subunits, and these subunits are monomers. So they're really, really small, and you will see what I mean later. So the four examples that we're going to study in this course is carbohydrates, lipids, proteins, and nucleic acid. So let's go ahead and start with nucleic acid. There's five different nucleic acid, and there's DNA. It's also separated into DNA and RNA, by the way, and it's made up of four repeating nucleotides. If you didn't know that earlier, and the only difference between these nucleic acids is the sugar that they use and the nitrogenous bases. So let's go ahead and look at these five different ones. So I want you guys to go ahead and take a couple seconds and look, see if you see any difference. So it's very hard. There are differences, but it's hard to notice. So the five that we're looking at right now is adenine, guanine, cytosine, thymine, and uracil. So uracil is we don't actually have it. It's used in bacteria, specifically in RNA. So if you can't see, the primary difference is the nitrogenous base, which is on the right. You can see here, this different from that. It's different from this, different from this, and different from that. And yeah, so it's very different. And then that's pretty much it. Now let's talk about carbohydrates. So there's a lot of different carbohydrates. There's the starch, which is the most regular one, most common seen. It's amylose, and that molecule can be broken down into glucose. And we eat starch. This come from, like, let's say potato and rice and all of that. We eat starch for energy. Glycogen also breaks down into glucose. We also use these for energy. However, these are like the meat from animals and stuff like that. And cellulose is the kind of the meat from plant. It's the carbohydrate from plants. We use this more for structure. Where we don't use it, but the plants use it for structure. Sucrose is glucose plus fructose, which is a sweetener that is much more sweeter than the regular sugar. And then lactose is glucose and galactose, which is in the milk products. So we could probably see somebody being, lactose intolerant, and they're like, once you give them milk, they start to vomit and have constipation and all that. Maltose is glucose plus glucose, which is also a sweetener. So let's go ahead and look at this. So this is amylose. These are just regular sugar joined together, and cellulose, I believe that's glycogen, is it not? And then all of that is a linkage and stuff like that. And this is what I was talking about. So this is how you join sugars together. So that essentially you're gonna take the water out and you're gonna join them together. So yeah, I was looking for that. So this is the condensation process. You remove water and it becomes this. And so let's talk about fats. So triglycerides are the major food type of fat. So it's made of glycerol and three fatty acid chain. It can be saturated or unsaturated fat. So we see it right here. So this is a glycerol. This is the thing that's in front of it. And these are the chains, the fatty acid chains. So it's this one chain of, well, I guess C carbon bonded to hydrogen. So this we're looking at an unsaturated fat. So by unsaturated fat, we mean that the carbons could actually attach to more hydrogens than it has already have. So right here, we can actually this carbon, these two carbons, it could actually break the double bond and have and get more hydrogen attached to it. So the main difference between saturated and unsaturated fat is just if there is a double bond. However, saturated fats is more found in animals and some plant-based sources, and unsaturated fat typically come from plant sources. Uh, Many people argue that I guess unsaturated fat is a lot more healthy than well saturated fat simply because we can actually digest it a lot better and it helps our cholesterol. So cholesterol is another form of fat. It produces ring structure. So as you can see right here, ring, ring, ring with the carbon chain. Now let's talk about proteins. Proteins are some very complex structures. They're made out of amino acids, which is their monomers. And there's from 80 amino acids to thousands. These proteins can be used for many functions because proteins are like this most important thing. It tells us what to do. It's like encoded in the DNA. So the DNA actually encodes for protein production, which is why DNA exists in the first place. So these proteins, they can command the structure, transportation, and enzymes. So let's go ahead and look at some protein. So protein are broken down into these amino acids. So amino acids are a pain in the butt to remember because there's a ton of them and they're grouped differently. So we actually don't have to remember this, but if you're taking med sci, life sci, health sci, not good. You're gonna remember all of this because they're different. So let's go ahead and look at some nutrition facts about different stuff, so we can see what's going on. So this is a liquid egg white. So we realize, hey, look, it's very healthy. It has proteins and a little bit of sugar and nothing else. So this is something you really want to eat, but not really because you want to eat this because it has a lot of protein. However, you're missing some fat. So usually it's a little important that you're actually taking a bit of fat. However, not too much. So gelatin, I guess if you guys know what it is, oh, I should have put a picture. So gelatin is like this little candy thing that is made out of it's made of animal collagens, which is skin, bones, stuff like that. It's like a little jelly that you eat. So this is okay. It has 6 grams of protein, no carbohydrates, some sodium, and no fats. So it's pretty good, pretty healthy. Veggie oil. So you have 10% saturated fats, and you have nothing else. So, but then you were like, look at this, unsaturated fat, mono unsaturated fat, and you're like, okay, that is pretty good because if you have more unsaturated fat than saturated fat. So usually unsaturated fat is better. If you use animal oil, there's going to be way more saturated fats. Then finally, we're gonna look at soft drinks, and you can tell how unhealthy this is. Even though there's no fat, there's a lot of sugar, and it's very hard for your body to digest and break down sugar. Well, in such high doses, and then we also have to talk about one of the most important things in soft drink that they don't actually listed on here: caffeine. You're not supposed to be ingesting that much caffeine when you're such a young age, especially those sports drink prime Red Bulls, or I guess what else is there? Monster. These ones are really damaging teens' brains, and it's not beneficial. So that is pretty much all I want to talk about in this class. I'm gonna I'm gonna edit in a really short thing that I want to add on to this. So prepare for that. Alright, so a little bit of add-ons. So we're gonna see how things are broken up at different time of our digestion. So in the mouth, we're gonna go ahead and intake. Let's say we took some carbohydrates. Let's say I took some bread. I'm gonna uh break down using amylase salary, and this is gonna break down into maltose. Break down into maltose. And then this maltose is gonna get into the stomach, and then the stomach is gonna do something. It's gonna inactivate amylase. It's gonna inactivate it, and then it's gonna get into small intestines, which is gonna break it down into monosaccharides, otherwise known as glucose. And then, so basically, you're breaking down maltose into glucose. Pretty simple. And then for protein, we cannot do any chemical breakdown in our mouth, so none. And we actually can't do anything to all of these as well, unfortunately, because we don't even have it yet. So in the stomach we have peptides. We're gonna break this into peptides. Break down using pepsin and HCl into peptides. This peptides is gonna get into the duodenum, and then we're gonna use protease. Break down using protease, which is trypsin and chymotrypsin, and this is gonna break down into uh, dipeptides. So this was polypeptides, by the way, polypeptides. And then finally, we're gonna do peptidase, which is gonna break down into amino acids, which is the simplest form. For nucleic acid, it's a little different. We can't do anything in the mouth either, but in the we also cannot do anything in the stomach, since this is only absorption. So this is majority only absorption, simply because, well, it's the center of most stuff, so only small intestine do its work. So we come in as triglycerides. Oh, sorry, DNA and RNA. So we're gonna use nucleases. So we're gonna use it to break down into well, I guess fragments of nucleic acid. So I'll say short nucleotide chains. And then we're gonna use something called using nucleosidases. Breaks down into break into nucleotides, ATCGU. Have fats. We do a little bit in the stomach. We have fatty acids. Fatty acids and plus diglyceride. So we're using lipase here. Using lipase. And then we're going to break it down into monoglycerides and simple fatty acid chains. And unfortunately, that's as far as we can go. Sometimes there's a little bit of glucose in it, and glucose. So yeah, that is pretty much all we have. And please remember this. And that's pretty much it. I'm gonna link some notes in the description. Please go check it out. And I will also link a gizmo that you should do. And that's pretty much it.